This is a lecture on photosynthesis, the carbon reactions, part 3. Please ensure that you have seen the previous lectures on photosynthesis, the carbon reactions, part 1 and 2 before viewing part 3. Please refer to chapter 8 of Tagues and Zeiger 2010, which is the recommended reading. This lecture is offered as part of the subject plant physiology, which is part of the Agricultural and Viticultural degrees offered at NMIT. My name is Dr. Nikki Cooley. In part 3 of the photosynthesis, the carbon reactions lectures, we will study the acclimation and partitioning of photosynthates and the formation of chloroplast starch, and we will also touch a little on sucrose. So let's start with a discussion on acclimation and partitioning of photosynthates. These are the products from the carbon fixation of photosynthesis. Eukaryotic organisms have to mobilize sugars from the site of synthesis, what we call source, to cells that use them for growth or energy, and we call these sinks. Arterioles of animals transport glucose. Conducting vessels of plants transport sucrose. Photosynthetic assimilation of carbon dioxide yields sucrose and starch as end products. Sucrose is synthesized in the cytosol, while starch is synthesized in the chloroplast. During the day, sucrose flows continuously through the plant while starch accumulates as dense granules in the chloroplast. The figure on the screen in front of you is from the recommended reading on plant physiology, figure 8.14. What it shows is the different structural components and where the different sugars involved are synthesized. You will see during the day that starch is a product of the Calvin-Benson cycle, as we've discussed in some detail. Also products of this cycle are the triose phosphates and the hexose phosphates, and these all go to produce the compound sucrose. Sucrose moves from the cytosol into the vascular tissues, where it is then transported through the plant in the vascular tissues or phloem. Sucrose can be used for growth, or it can be converted into starch and fructanes and used for long-term carbohydrate storage. In the cytosol of the chloroplast, starch is produced. It can be broken down into glucose or maltose. This can also be broken down into sucrose, which can again be moved into the vascular tissues. Starch is a very important component as it is the main storage carbohydrate in plants. It is surpassed only by cellulose in abundance. Starch is a complex polymer made up of two components, amylose and amylopectin. Starch is called a polysaccharide. That means it has many of the glucose units. The glucose molecules can be arranged in many different fashions depending on the bonds between each glucose, and these bonds are known as the glycosylic bonds. This is what causes some of the differences between amylose and amylopectin. Amylose consists of approximately 500 to 20,000 glucose units, whilst amylopectin has approximately 10 to the 6 glucose units. In the figures at the bottom of the slide, you can see a demonstration on the left-hand side of starch. This is demonstrating 3 glucose molecules bound together. On the right-hand side, you can see some modelling of some of the different shapes that starch can be constructed of. At night, chloroplast starch can be broken down to maltose, the main form of carbon exported from the chloroplast. For normal mobilization of linear soluble oligosaccharides, oligosaccharides means many saccharides, starch must be first phosphorylated prior to hydrolysis. Maltose is exported from the chloroplast and into the cytoplasm and converted into glucose. The figure on the slide is from the textbook, figure 8.17, and this illustrates the concepts we've been describing above. Another and very important molecule of photosynthetic assimilates is sucrose. Sucrose biosynthesis is described. The triose phosphates from the carbon-Benson cycle, including glycosylceramide 3-phosphate and dihydroxyacetone phosphate, are transported from the chloroplast to the cytosol. In the cytosol, triose phosphates are converted to fructose and glucose monosaccharides. The sucrose 6-phosphate synthase enzymes will catalyze the formation of sucrose from the molecules fructose and glucose. In addition to proving carbon for growth and polysaccharide biosynthesis, sucrose also acts as a signaling molecule. For agricultural perspectives, sucrose is probably the most common sugar to be transported around the plant. The image on the slide shows sucrose biosynthesis. I will not expect you to understand the details of this, other than that sucrose is compiled of glucose and fructose molecules. The production of sucrose, like so many of these reactions, involves the input of energy in the form of ATP. Inorganic phosphates are transferred. I would recommend watching the following video on photosynthesis and respiration, as it sums up many of the concepts that we have dealt in the 3 phases of the lectures here. This will assist with your learning of this subject. That concludes part 3 of photosynthesis, the carbon reactions. I hope that you have understood this introduction to assimilate partitioning. We will deal with this concept further in the tutorials, and we will examine some scientific papers to support our understanding. I hope that you have understood the role that starch plays in assimilate partitioning and the processes of starch and sucrose biosynthesis. I do not expect you to remember every single molecule in starch and sucrose biosynthesis, but I do hope that you have an understanding of the overview. Thank you very much for taking the time to listen to these lectures.